I'd like to briefly recognize our board members on the call. I see Carmel Rafalowski and our president, Ann Corval, and a very special thank you to Cindi, Brandt, Levin. Cindi is the FDMAS Alliance's vice president and the leader of Team FDMAS in the UPenn Orphan Disease Center's $1 Million Bike Ride. She's championed this initiative since 2014, and beyond the $1 Million Bike Ride, she's also devotes significant time and energy to the organization and our mission, and we're very grateful to have her leadership and determination. At this point, I am going to turn it over to Cindi to lead us through the next.
Thank you. Thank you all for being here. It's a privilege to help host this seminar with everyone that's participating, presenting, the researchers that are here. As Tova just explained, I have been involved in this organization since 2014, before the $1 Million Bike Ride started. And at that point in time, we didn't have a lot of research except for NIH. And I was thrilled to get involved with UPenn and the rare disease organizations that they support. And it is going to be 10 years this year in June, which is very exciting. And we have raised $1.2 million in research to date with 16 publications and counting. And today we'll hear some of the reports of the 2021 awardees. And I first want to present a video that Tova had prepared, which sums up these past 10 years of our $1 Million Bike Ride. Thank you, Tova. Fabulous, Tova. I'm sure everyone here. Am I still on, Tova?
I can introduce our moderator for today, Dr. Michael Collins, is senior investigator in the skeletal disorders and mineral homeostasis section in the National Institute of Dental and Craniofacial Research. And he's devoted his career to the study of FDMAS, including close collaborations with this organization since our founding. He served as the FDMAS Alliance's medical advisory chair for over a decade and is currently acting as scientific advisory chair. We're very grateful to you, Mike. And from here, I'll let you take it away.
Thank you, Tova. Thank you for the introduction. And especially, Cindi, thank you and Tova for the nice video you put together. But mostly, thank you for the dedication and time that you've put towards this, which has really made a huge difference. You've expanded with this money, expanded the scope of people, investigators interested in and pushing this forward. This seed money from the $1 Million Bike Ride has resulted in NIH grants, Department of Defense grants. I just think it's been a huge success. And thanks a lot from us and everyone involved. I'll move right ahead to the first presentation, which is by Biagio Palmisano. Tova, maybe you could mute the person who's talking. Anyway, so I've known Biagio for some time. He works in Mara Romanucci's lab in Rome, where previously Paolo Bianco was the leader of that lab, which has really done seminal, important work in this field. Biagio is a postdoctoral fellow there at Sapienza University in Rome, and his research interests focus on the biology and pathology of bone marrow stroma, with a specific interest in the pathogenesis of fibrous dysplasia, and he's going to present to us his work in this area for which he was funded by the $1 Million Bike Ride. Take it away, Biagio.
Thank you very much, Mike. I'll try to share my slides. Okay. Hello, everyone. It is a real pleasure to be here and to present updates from this project that was funded by MBBR FD Mass in 2021. I'm a basic researcher, and the work I'm presenting today was entirely performed on mice, my little friends. So FD, fibrous dysplasia, is a unique example of skeletal disease caused by the dysfunction of the cells that give rise to the skeletal tissues, also known as the skeletal stem cells. And this leads to the accumulation of fibroblast-like cells in the bone marrow of affected skeletal segments. This so-formed fibrous tissue release high level of rank ligand, therefore stimulating bone resorption. And we know now that inhibiting bone resorption is the key to effectively treat the disease. However, this long-standing concept that fibrous dysplasia is a stem cell disease has remained largely unexplored from a research point of view. And we know today that there are many cell populations with skeletal stem cell properties in the bone marrow. And this project arose from some important observations that were done in our lab during the years and are reported in these two articles that we have published. The first shows that inhibiting rank ligand is an effective therapy for fibrous dysplasia. We demonstrated this in mice, but now there are data in humans that demonstrate the same. The second reports the identification of skeletal stromal cell population in the mouse bone marrow that can be identified by the AdipoQ expression. That's why hereinafter we'll call it AdipoQ stromal cell. And we, another group, have demonstrated that these cells give rise to the osteoblasts, so the bone-forming cells. So it has skeletal stem cell properties, but also stimulates the bone resorption by producing rank ligand and other osteoclastogenic factors. How did we study the cell population? So we generated a mouse model where the cell population express a green fluorescent protein so that it appears green when we analyze histologically the tissues of the mice. And for example, in this picture, the AdipoQ stromal cells are found around this bone trabecula that is generated by these cells. They are found forming this spider web among hematopoietic cells surrounding the blood vessels in the bone marrow. But importantly, we found that these cells also around intraosseous blood vessels, where, by the way, FD likely starts. So in our mind, this cell that appears postnatally exactly like fibrous dysplasia was a perfect candidate to take part into the pathogenic process of FD development. So to understand how the cell population could be linked to FD, we generated a murine model where the GS-alpha R201C mutation the causative mutation of fibrous dysplasia, is selectively expressed by adiponectin stromal cells. When analyzing the bone of these mice, we observed the formation of osteolytic lesions that were filled by a fibrous tissue expressing rank ligand with a very high number of osteoclasts, similar to FD lesions. According that adipocustromal cells had the green fluorescent protein, so we could see their localization. And surprisingly, we observed that the fibrous tissue was not composed of green mutated osteogenic cells only, but it contained other non-mutated cells of different origin, exactly like the mosaicism that we see in human lesions. These results supported the role of adiponectin stromal cells in the pathogenesis of the disease, especially in the initiation of the FD bone lesions through the stimulation of bone resorption. So we developed this project with the general goal to better characterize this cell population in the context of well-established FD lesions, but also to understand its involvement in the bone resorption. The project is still ongoing, and as the two specific aims you can see here, the first aim wants to understand how adiponectin stromal cells behave in a context of a well-established FD lesions where all the other cells are also mutated. We then took a mouse model of FD that develops lesions in almost all the skeletal sites, and we modify it a little bit genetically by, again, expressing GFP in adiponectin stromal cells. The resulting mouse model developed FD lesions in many skeletal segments, and here we can see the tibia and the femurs that are affected, but also the spine. And this is the picture, this is a confocal microscopy picture of the mouse femur. On the left, we have the wild type, and on the right, we have the femur from our new FD mouse model. And we can see that there are FD lesions here, and there is something green expressing GFP in the trabecula. When zooming on this mouse FD lesions, we saw that osteoblasts surrounding the bone trabeculae, but also osteoblasts surrounding the fibrous tissue were green, as we were expecting, but the cells in the fibrous tissue were not, suggesting that they do not derive from adipocustromal cells, and so this suggests also that the fibrous tissue may not be the osteogenic tissue that we believe it was. Then, to understand the role of this cell population and the bone resorption in initiating FD bone lesions, we genetically removed the rank ligand from this cell population and assessed the effects in a mouse model of fibrous dysplasia. To do this, we took advantage again of mouse genetics, and we created a mouse that developed FD, but have the specific deletion of rank ligand only in adiponectin stromal cells. I will refer to this mouse model as triple transgenic in the next slides. Radiographic results reveal that the triple transgenic mouse grew normally and did not show shortening of the skeletal segments that is usually an adverse effect that can be seen when rank ligand is inhibited systemically. Moreover, while FD mice on the left developed bone lesions in the femur, the triple transgenic did not. So we performed a histological analysis that confirmed the almost null presence of FD lesional tissue in the triple transgenic mice. The quantification of the fibrous tissue revealed a statistically significant reduction of the compared to FD mice. So with these experiments, we were able to prevent FD development by removing rank ligand only in a specific stromal cell subset in the bone marrow. In conclusions, we have identified a postnatal skeletal stem cell that is involved in the pathogenesis of FD in mice. These cells do not form the fibrous tissue, but we already know that drives the FD development by stimulating bone resorption by rank ligand expression. And we know that if we inhibit rank ligand in these cells, we can prevent the appearance of the disease. What's next? So we want to develop a therapeutic approach to inhibit the bone resorption activity of these cells, because inhibiting these cells only does not affect cartilage growth and therefore do not interfere with skeletal growth. Denosumab, unfortunately, acts also on cartilage growth plate, inhibiting bone resorption at those sites. Importantly, these cells have been recently reported also in humans. There are some data sets in single-cell RNA-seq demonstrating that these cells are also present in human bone marrow. So more studies are needed to better characterize them in the context of fibrous dysplasia. And with this, I want to thank my collaborators at Sapienza University of Rome and all of you for the attention, and I'm happy to take questions.
Thank you, Biagio. That was impressive and very clear. Tova, how do you want to do questions? Will people just, do you want them to raise their hands or
You know, that's entirely up to you, Mike. I mean, the chat might be a nice way for folks. But also I think if folks are comfortable with hand raising and coming on and asking on video, that could be nice too. So.
Okay. And Tova, how much time do we have for questions right now?
Let's do, you know, maybe keep it to 5 minutes. So maybe 2 questions if we.
Okay, sounds good. And is there anyone with questions for Biagio? And while they are being formulated, I'll ask you a question, Biagio. It looked like that the triple transgenic in the slide that you showed of the histological sections of the bone, it looked like it actually had high bone mass. I didn't see a control for that, but is it dramatically high? Is it like osteopetrosis high? It looked like it had bone marrow. It was fine in that way, but I was just curious.
So, of course, I have the controls there for the sake of, you know, simplicity. I took away the other controls. So I have the control that does not have the GS alpha mutation, but is only the adiponectin rank ligand flox. And yes, this mouse developed a high bone mass phenotype. What was interesting is that in the when you leave the mouse, the adiponectin rank ligand flox mouse develops a high bone mass phenotype. But when you also put the GS alpha R201C mutation, you still see the high bone mass. It is a little bit, let's say, reduced compared to that control, but it's still, there is a high trabecular bone mass. I would not say osteopetrosis. The cartilage thickness was normal, and there was bone marrow there. There was enough bone marrow. And also when you check rank ligand, in total rank ligand levels in that mouse, you do not completely remove rank ligand from, and you do not completely remove osteoclasts from those skeletal segments. So I would say high trabecular bone mass, yes.
And if there are no other questions from anyone else, I'll ask a second question, which is, what's the phenotype in the craniofacial bones of both the adiponectin cells that are expressing the mutant GS and in the triple transgenic? Have you looked at the craniofacial bones in that animal?
Yes. So our classic mouse model, so our canonical mouse model, I mean the F1 alpha, develops lesions, FD lesions in jaw bones, in calvaria, in all the skull. And I haven't checked the triple transgenic, but I remember that from a radiographic analysis I did, I could not see abnormalities in the craniofacial bone, if rancligin inhibited there was causing something. But I have to check that. I have to check it by then.
Who's that? Where are you gonna look at?
Looks like there's Kelly. Kelly, did you have a question?
No, we just got to log on.
It looks like you're in a hospital.
Yeah, in the ER, you know.
Sorry. Sorry. All right, well, if there are no other questions for Biagio, we will move on to the second presentation. Is that okay, Tova?
Absolutely.
And so this is a joint presentation by two investigators who have a collaborative project, which again, I think is really great and really reflects the overall collaborative nature between the people working in this field, which is not always the case. So it's really nice to see. So the two people who are presenting are Dr. Charlie Hoffman and Dr. Fernando Fierro. Charlie is a PhD at Boston College. He got his bachelor's at MIT and his PhD from Tufts. He did graduate work and studies at Harvard Medical School as well. And he's been at Boston College since 1990. Fernando is at the University of California, Davis, where he's an associate adjunct professor. And his interest is in stem cells, at both their basic biology and the application and the development of novel cell-based therapies. And they have a really nice joint project that I think it'll be fun to listen to. So with that, take it away, Charlie and Fernando.
Thank you, Mike. I'm going to share my screen, I think. Let's see. Okay, thank you, and thank you to the organizers for putting this together. I do want to take a quick minute or second to comment that this collaboration actually grew out of a previous one of these update meetings from, was it the 2019 recipients, I think. And that's where Fernando and I first met and started this collaboration. So I am a yeast geneticist, not the type of person you might expect to hear from, but my lab has been doing chemical genetics, where we genetically modify fission yeast for small molecule screening, and identified a collection of compounds that lower cyclic AMP production by the target of GNAS, these transmembrane adenyl cyclases. So I'm going to try to go fairly quickly through this. So as you know, fibrous dysplasia, McCune-Albright syndrome, is caused by mutations in GNAS. This protein gets locked into an active conformation that stimulates this class of enzymes called transmembrane adenyl cyclases. There are 9 different genes that encode these in the human genome, and they produce the signaling molecule cyclic AMP. And so this is now creating an overproduction of cyclic AMP. We've engineered fission yeast strains to express these mammalian cyclases together with a mutationally activated human GNAS. It was a gift from Mike, actually, and carried out screens for small molecules that could lower cyclic AMP production, screening about 125,000 compounds using 2 different adenyl cyclases. And we came across this group of 5 molecules that were structurally related and could lower the cyclic AMP production by these yeast strains and also by human cell lines. We have since shown with funding from the Alliance that these compounds lower cyclic AMP production in our yeast strains in the absence of GNAS, so they're not simply blocking GNAS-mediated stimulation of the cyclases. However, a direct biochemical assay of transmembrane cyclase activity fails to show these compounds working, and that's generally the type of assay needed to get a medicinal chemist interested in what you're doing. So we have this question of whether or not they act directly or through an intermediate target. And so that's been the focus of our recent work, trying to figure this out. We have reason to think it's direct, and therefore we're orienting our studies trying to address that question. So it's important to understand what the structure of an adenylyl cyclase looks like. For this talk, there are 12 transmembrane domain passes that hold two different cytoplasmic domains from different parts of the protein together to create the active site. Upon activation, GNAS specifically binds to the C2 domain. So this is cytoplasmic 1, cytoplasmic 2. And so what might we look for in the absence of an in vitro enzyme assay that can support direct activity? Well, we're taking a few approaches. One is to construct chimeric proteins. And what that means is a protein that's composed of a portion of one of the transmembrane cyclases and a portion of another one. So what we've done is taken the C2 domain between cyclases 4 and 6 and swapped them out into the two reciprocal cyclases. We're in the process of doing a similar swap with the cyclase or cytoplasmic 1 domains. And I'll show you a little data in a minute from this work. A second idea is that if we look at active compounds against a panel of native cyclases, so not these chimeras, but just 6 different cyclases for which we can easily measure cyclic AMP production, one might expect that if they're working directly, we will see distinct patterns of potency against the different cyclases. Whereas if they're working through an intermediate protein or process, then the patterns should sort of mimic each other, where the most potent compound is still the most potent against all of them, and the least potent is the least potent against all of them, and nothing very selective looking. And we're in the progress of doing that work. And finally, there's a model for how these compounds might be binding, and this model would involve an interaction with both the C1 and C2 domains. We've constructed 4 single amino acid changes in the putative active site or binding site. We see very little effect of these mutations, so we're in the process of making 2 mutations at a time to see if that has more of an impact. So here's possibly one of the most exciting data I have to show right now, which is that if we take the C2 domain of cyclase 6 and swap it into cyclase 4, we do not alter the potency of this compound BCAC53. And similarly, if we take the C2 domain of cyclase 4 and swap it into cyclase 6, we see similar response. So this would argue that the C2 domain, which is where GNAS binds, is not the direct target of these compounds, and that that somewhere else in the protein is where they're interacting. Similarly, if we start looking at individual cyclases, and this is a different one of our compounds, BCA63, and we assess this at the concentration required to inhibit 50% of the activity. I'm sorry, I had not labeled this axis. So this is percent inhibition. So the higher the value, the more inhibition you see. So as we increase the concentration of compounds, we get greater inhibition, but the amount of compound needed to inhibit 50% of the cyclase 4 activity is only about 1.15 micromolar versus 2.5 micromolar for cyclase 5, and cyclases 6 and 9 are sort of intermediate sensitivity. So just a quick recap, the compounds are working on the cyclases in the absence of GNAS, so it either inhibits basal activity. Again, it could be intermediate, but we think that's unlikely. They don't appear to work through the C2 domain, which is where GNAS binds, and we are continuing our work on mutating and altering these cyclases and also profiling the native cyclases against a panel of 12 out of 12 compounds that show potency out of 44 structurally related compounds. So quick, and then I'll hand this off to Fernando. Quick thank you to the people in my lab who did the work, our collaborators and people who provide us with clones and funding from $1 Million Bike Ride. So I hand it over to Fernando now.
No. How's that? You see my screen?
Looks
Yeah,
good.
great.
Mhm.
Thanks, Charlie. And thank you all for being here. As Charlie mentioned, this is for me a great opportunity. I love this updates because of course it kind of keeps us with a cable to the ground, you know, like just what are we doing, how far are we on it, and so on. So it's great to be here to hear what the others are doing, and of course that way we keep fostering our collaboration. I'm currently working on an MDBR grant with Charlie, of course, that's what I'll be presenting you. But I've also, in the 2021 MDBR, it was a collaboration with the group in UCSF, Kelly Wentworth and Ed Xiao. And so Dr. Wentworth, I think she's presenting after me, she will be talking about what we are currently still doing on the single cell data. Every time we hear about a patient undergoing surgery, we go and try to get a bone specimen and look at the cells there using this single cell transcriptomics technology. But now I'm going to focus on the model that we use to test the compounds that Dr. Hoffman has been identifying. So the first, I'm going to do this very simple and basic. The first question is, what cells do we use? And these are what we call bone marrow stromal cells. Some people call them mesenchymal stem cells. They have different names, but what they really are simply, I'm not sure if you see my pointer here. So any person can donate bone marrow, and that's typically done by taking an aspirate from the upper iliac crest, from the hip bone. It's just an aspirate from the marrow, and we do a small centrifugation just to get rid of mostly the red blood cells. And when you put that into a tissue culture flask, of course, it will contain a mix of different cell types. But most of those cells are non-adherent cells, so they are washed off over time. And the cells that are attaching and that resemble these fibroblasts, so to say, are these longer cells, they start to proliferate very well. And eventually, let me see if I can. No, I think I can play this video. But, well, they become a very homogeneous cell type. And this is what we called MSCs, bone marrow stroma cells. These cells have likely a lot to do with the adipose-Q cells that were just introduced. So these cells are our model. Why? Because these cells have clearly very good osteogenic potential, and it's thought to be very closely related to the cells originating FT lesion. So what we do to model the disease is to take these cells and now use a lentivirus. So the cells, again, are derived from just a healthy donor, or actually multiple healthy donors. Each experiment we do is done with cells from a different person. And the conditions that are compared are, of course, always within the cells of that same person, either transduced with just a control lentivirus. This is a tool we use to modify the cells genetically. It makes the cells glow green, the same GFP that was also introduced earlier, or wild type GS alpha, or the mutant R201C, R201H. And as expected, the cells carrying the mutation have higher cyclic AMP levels, Although I must say that this outcome measurement is critical for us, the cyclic AMP, because the goal is to test how the compounds will be able to reverse this, right? Just as what was shown in yeast, we want to see them also in our human cells to bring those values back to normal levels. And for that, we are still optimizing this method of measuring cyclic AMP, because we noted that at least 2 products that are available to do these measurements have significant flaws that can lead to misleading results. So we are on the process of tweaking them and coming up with our own perfect protocol, or better at least, to be very quantitative about it. What are those nanomoles or micromolar concentrations of cyclic AMP in each cell? Another clear characteristic of these cells is that the cells carrying the mutant GNAS have a distinct morphology. Right away, about a week after having been transduced, the cells don't look anymore like the spindle-shaped fibroblasts, but rather become these cells that remind me of a spaceship of Star Wars. But this is typical, actually, morphology for cells accumulating high levels of cyclic AMP. You can induce that chemically, and that would also induce the cells to have that shape. These cells have very low migration. I want to go fairly fast through this data because I presented it as a poster in our meeting in September of last year. And so I want to focus more on what we are currently doing with them. Yeah. And the most important, these cells have reduced bone formation as measured by enzymatic reaction called alkaline phosphatase activity. This is a marker of pre-osteoblast. It's reduced in the cells with the mutant GS-alpha, and the mineralization too. And the proliferation is also reduced in these cells. Yeah, so they have cyclic AMP. This is just a quick summary of what I already had shown to many of you before. The distinct morphology, impaired cell motility, and impaired bone and proliferation, which sounds counterintuitive, right? Because FD is characterized by rather a large bone mass and high proliferation. So we are definitely missing something. To get into that, I will now switch from paying attention to the autocrine effect, what happens to the cells themselves because of carrying the mutation, to what we call a paracrine effect, which is these cells that carry the mutation are secreting factors that could be affecting cells that don't have any mutation. They're completely normal cells, but are exposed to these cells, just like it was recently shown the presentation before from Biagio, beautifully shown how the fibrosis was there, although it wasn't made by the cells carrying from the ADCOQ cells. So this is very much in that line, right? The effect of the paracrine signaling. And that is based, of course, in previous work from the group in Rome, which I think it's critical. That basically can be summarized that these same cells, these also derived from human people, where FT patients or healthy donors, they were injected into mice, immune deficient mice, so that the cells would not be rejected. The important outcome is that mutant cells only carrying mutant GS-alpha do not make FD. They actually die off. They cannot make it. Of course, if you just inject healthy cells, you get normal bone. There is not a problem. So it was suggesting this very now classic work that it's the combination of mutant cells and non-mutant cells that are required to generate an FD phenotype. So that's the basic, this is the scientific premise that made us want to study what happens to the factors with cells that are non-FD cells, but exposed to FD cells. And for that, we use this. One of the models is this. This is what's called a transwell assay. We put in the bottom of this insert normal MSCs, not modified in any way. And here are the cells with one of the 4 constructs that I showed you before. And this membrane has a pore that is small enough that it wouldn't let the cells go through, but the soluble factors, for example, small proteins and so on, can easily diffuse between the 2 cell types. And to our surprise, these cells, now normal cells, exposed to cells that were carrying the mutant FD, actually show increased bone formation and increased proliferation. So strongly suggesting this is now very important to us, suggesting that the cells are secreting something, the mutant FT cells with mutant GS alpha are secreting something that is pro-proliferation and pro-bone formation. This is just summarized here. This needs to be repeated because you see that we have only significance for one of the GS alpha, but obviously this is a common effect. So we are just in the process of consolidating this data, but we are quite confident about it. To have a first hint of what is that factor, we did what's called a phosphoarray. It's a small membrane. It's basically a biochemical assay filled with 42 different antibodies that are going to tell us about what signaling pathways are being activated because of being exposed to in this case, it was just the supernatant of the cells carrying either wild type GS alpha or the mutant GS alpha. And to our surprise, the main pathways were really only 3. Wind, which I think it's not new to many of us. We knew that wind was exacerbated, and that could be a cause. We see increased phosphorylation of beta-catenin, for example, with the mutant. CREB pathway, which is very interesting because this is the cyclic AMP pathway, which means that the mutant cells are likely secreting cyclic AMP that is acting not only as a secondary messenger, but as a primary messenger, again, on normal cells. We are activating cyclic AMP pathway on cells that are not carrying the mutation. I hope I've been clear about the cells that we are interrogating are just normal cells. They are, however, exposed to the cells carrying the different mutations. This pathway, which I'm not familiar at all with, WNK1, it's highly associated with angiogenesis, actually. It stands for without lysine, with no lysine 1. And we need to explore this, of course, further. And then to a lower extent, there were 2 more pathways activated too. One is ERK1/2, which is famously associated with proliferation. So this could be maybe the culprit for why the cells are growing more when they're exposed to the mutant cells. And STAT3 that is also critical to osteogenesis and so on. So that's where we are right now. The wind data, I can show you this again, but basically we have been confirming now, we are going through the process of confirming the activation of these pathways using Western blot or flow cytometry. The activation of wind is, as I said, already known to many of us, and that was also, it's also shown here. So current experiments, as I mentioned to you at the beginning, we still need to optimize cyclic AMP measurements because this is key for the compounds we will be testing, right? That's our first a tool to determine which are the best candidates to move forward. And I should mention here that the candidates are from 2 sources. One is, of course, Charlie Hoffman, because of the screening that he did. But I'm also testing a few, I think 3 or 4 compounds coming from Dr. Wentworth, because they used in silico algorithm, a computer modeling to determine what would be a good molecule to inhibit GS alpha hyperactivity. So I will be testing those compounds too in our system. Yeah, and that's what we need to do here in this second aim. And then, of course, once we have ideal candidates, either because they are active in lower concentration or because they can have an effect on only mutant but not wild type cells, there are many criteria to pick our favorites. We will be doing a lot of these other assays that I've just shown you to see how much of that can be reversed thanks to having added the inhibitor, for example, the changes in morphology, the changes in the paracrine signaling, and so on. And then finally, we also want to move this forward towards testing them in mice. So we will be doing some very similar experiments to what I showed you had been done by the group in Rome a long time ago, but now with our genetically modified MSCs, of course, combined with normal MSCs, since we see that there is a requirement for both to be there, and then there test the compounds. So that would be, of course, now just hopefully 1 or 2 leading compounds. We don't want to go through all the compounds in all the mice. That would be a very too big of an experiment. And that's it. This is my group. I really feel very thankful for the FDMAS Alliance and for the funds they have raised through the $1 Million Bike Ride program. And here, my collaborators, I think I've mentioned them a lot, so you know that I'm a big fan of them. And that's it. Yeah, these are the people in the lab that have been mostly working on this project. So thank you all. And I think we are now, Charlie, happy to take questions, right?
Thank you, Fernando.
Yeah.
And Charlie. Are there questions for either Fernando or Charlie from anyone at this point? Lillian has her hand up, and Adrian had. I'll let Adrian go first because she had a question in the chat, and Adrian's question in the chat was, I guess this is for maybe any of you guys. Is there a role for AI to advance this study? If so, how? I guess that's probably for you, Charlie and Fernando.
Well, certainly AI is being used now to accelerate drug discovery. That hasn't been something we've dabbled in yet, but that does seem to be where things are headed.
Yeah, totally. I agree. And some of that is, of course, the basis of how Dr. Wentworth came to discover their key molecules, right? It's about modeling the protein with or without the mutation to understand exactly what's the conformational change, and then how these small molecules will be hopefully docking to just the cells that have the mutation and not to the other cells. So these algorithms are still improving. I heard it just came out a new version of the what's called the super protein modeler now that has an amazing prediction ability compared to what's done experimentally. They come to something extremely close to that. So of course that would be, we will be using that a lot, yeah, to get to the best compounds.
So I think the simple answer to your question, Adrian, is yes, and it's being used and it's making a huge difference. Lillian, you had your hand up, but it's down now.
Yeah, no, I wanted to keep it up. So I have a question for Fernando. That was a great talk. So if I understood correctly, you said that you think that cyclic AMP is coming from the mutant cell going to the wild type cell. Do you have, are there any other molecules? You have STAT3, have WNT being activated, but do you know whether these mutant cells are releasing WNT ligands or IL-6 or any other?
Yeah, I don't think there are secrets now. We know that there is a WNT4, especially WNT5A is a main WNT protein that is being secreted. It's already highly expressed in these cells, but it's still doubled because of the mutation. So that makes the levels extremely high. Now, the paradox is that WNT5A is considered what's called a non-canonical WNT protein, and that, but we see a lot of canonical WNT being activated, so that could be because of other WNTs, like a WNT4. We're not sure about how that works. Yeah. The part of the cyclic AMP is critical. It's actually, we can detect the cyclic AMP in the supernatant too. Of the cells carrying the mutation, you don't even touch the cells. You just take what's being secreted, and there is the cyclic AMP, and it's elevated in the cells with the mutant GS alpha. So how it's entering the other cells, we don't know. And but we see the pathway being activated. Of course, we should also measure directly if the cyclic AMP is increased in those cells. That's something we still need to do. Yeah.
Thank you.
The Hans has his hand up.
Hans Smithak again. Well, if you think about giving a compound that inhibits cyclic AMP, what effects do you expect in non-FD tissues? So, I think important question.
Yeah, that's a great question that I'm not perhaps the best person to answer, but we always aim to just modulate levels. Of course, to completely inhibit cyclic AMP production in all cells would not be viable. We need that signaling pathway. So there needs to be a way we determine what's the right amount of compound to just avoid that excess of cyclic AMP that is happening in some cells. without hopefully altering too much others. I think there is a large group of chemical compounds that act on inhibiting GPCR signaling, this whole signaling cascade. And of course, it's all about the dose so that they don't become, so to say, toxic to all the other cells. So that's a critical question, of course, that we need to address. How can we use it? Perhaps there is a way to even administer the compounds locally in the lesion instead of just giving that to the patient as a medicine. Those are questions that we will be hopefully addressing once we are starting to see if they are having these side effects or not, if we can make them specific enough to only the cells carrying that excess of cyclic AMP and so on. So that's a great question, though. We need to.
So there actually are a couple other questions, but it's 7 minutes of, and we wanted to finish by 1:15, and Kelly Wentworth has yet to present. So if we have time when Kelly's finished, we can come back to Kelly Cohen and Cindy's questions. So with that, I'll introduce Kelly Wentworth. Kelly Wentworth is at the University of California, San Francisco. She's an endocrinologist in the Department of Medicine, and her research focuses on understanding the role of GS protein-coupled receptor signaling in skeletal development, with a focused interest in craniofacial fibrous dysplasia of the bone and the McCune-Albright syndrome. So Kelly, it's all yours.
Thank you, Mike, for that introduction. Sorry, I'm clicking around on the screen here. So today I am representing both me and Ed Shao, both of us here at UCSF. And for the first part, the first 5 or 8 minutes, we'll talk about the work that we've been doing funded by the MDBR that's focusing on identification and signaling characteristics of the GNAS R201CH selective inhibitors for FD-MAS. And I think this really builds nicely on Charlie and Fernando's talk, because this also uses some of the AI compounds. So I'll skip through this. This is just a basic background in case there was anyone on the call that wasn't as familiar with fibrous dysplasia McCune-Albright syndrome. But I think we are all familiar with the physical characteristics of this disease and the important fact that as the science advances, we're getting closer to developing medical therapies for this disease. But as it stands right now, there are no curative medical or surgical therapies. And so we're constantly seeking the opportunity to potentially develop new therapeutic options. And so the GNAS mutations, which are the causative mutations in fibrous dysplasia, are really acquired in early embryogenesis and can affect multiple different cell lineages, as is depicted here. And that's what leads to the wide phenotypic spectrum of this disease. And importantly, in fibrous dysplasia, it is the causative mutation affects the GS alpha protein. And the GS alpha protein is a G protein, and it is responsible for transducing or carrying the signal of different hormones and other chemical signals that happen in the cells from the outside of the cell to the inside of the cell. And typically, the GS alpha protein then activates adenylate cyclase and increases cyclic AMP and protein kinase A and downstream phosphorylation pathways. In fibrous dysplasia of the bone, what happens is this mutation in GS alpha prevents the protein from actually turning off, and so it just constantly signals either on its own or after it's been stimulated by a ligand. And that's how you get all of the different potential effects in different cell types. So importantly, this raises the question, is it possible to find a drug that can target the GS alpha protein and stop this overactive pathway? So G protein coupled receptor signaling is critical in all tissues. So unfortunately, just identifying one drug that can block the GS alpha protein is not a good approach because it would have widespread effect. And I think that's one question that someone had raised. What happens if you just target this protein? Well, it would have significant effect on other tissues. So one potential approach is to target the actual mutated protein itself, so the GS alpha R201H or R201C protein, and try to avoid having a compound that would also affect the wild type. And so that's what we have been working on. So several years ago, we collaborated with a company known as Atomwise to work. And what they are is they create this this network where they use this AI computational approach, and they screen large libraries of compound structures and like over 1 million different compounds that are screened. And what they do is they take the well-known published, protein of GNAS wild type and then R201H, the protein scaffold, and they screen their large library of molecules to see if there are compounds that might be identified that could be predicted to bind to and affect the R201H or C. The screen was actually done based on R201H, not R201C, but we thought, perhaps there's, similar enough structure that it could inhibit both. The goal was to identify compounds that could bind to and ideally inhibit the GNAS R201H, but not the wild type. And so out of that screen, 71 different compounds were selected, and we used our human iPS cell model to test these compounds in vitro. And so our initial screen was performed in these cells, and we took 50 micromolar of each of the 71 compounds and incubated it with our cells. And we measured the cyclic AMP levels in cells that were not treated and cells that were treated. And we used both wild type cells and then cells that had been mutated so that they contained the R201H mutation in the endogenous GNAS locus. And similar to what Dr. Fierro had described, we use the Promega CampGlow assay, which is a relatively simple assay. It's a luciferase-based assay. And so when the G protein is active in its active state, it activates adenylyte cyclase, and then cyclic AMP levels are increased in the cell. And the high levels of cyclic AMP then activate protein kinase A. And protein kinase A, one of its jobs is to phosphorylate different substrates. And so when cyclic AMP levels are high and protein kinase A is active, it converts substrates into a phosphorylated substrate. And as part of that process, ATP is used up. And ATP can be directly measured in this cell because it can interact with the luciferase enzyme and produce light, and that light can be measured. So when ATP levels are high, light is given off. However, it can be a little bit complicated to kind of look at this, but one thing to remember is that when you have high levels of light or luminescence, that actually corresponds to low cyclic AMP levels. So for our initial screen, I'm not showing you all 71 compounds, but we screened all 71 compounds in triplicate in our R201H iPSC model. And we had identified several different compounds that potentially were promising candidates for future testing, some of which had similar activity in both of the wild type and the R201H cells, some of which had kind of inverse relationship, which was what we were looking for, and some that seemed to activate. Because these compounds were not predicted to just inhibit the protein, it was also possible that these could activate the protein. So based on this, and I should mention that this was part of the Atomwise AIMS program. It was part of 318 other groups that were studying, that used their network to generate compounds using AI. And this was just published, actually, I think a couple days ago in Nature Scientific Reports. And so this was kind of an exciting way of seeing how AI can be used to identify compounds. And if you have a good system for testing that, then these compounds can really potentially be used for drug development. Our compounds weren't listed as an obvious hit, but just were selected for promising future testing. So we then took 10 candidate compounds and asked the question, do we think that they can also modulate GNAS R201C signaling? And to test this, we used SW48 cells, and these are human colon cancer cell lines that carry the R201C mutation. And we performed the same assays in wild type cells, wild type SW48 cells, and then the SW48 cells that had the R201C mutation. And from this, just to sort of briefly show the results, each dot here represents a triplicate, so a replicate of 3. And we were looking at levels of cyclic AMP after treatment. And then the black is the wild type cells, and the red are the GNAS R201C SW48 cells. And a couple things to point out. Some of these compounds did look like perhaps they had more activity in the GNAS cells. For the ones where we had 3 different dots here, that represents triplicates, but also biological replicates, where we had grown these cells at different time points and performed them on different assays at different time points. Some of which, looks not that different from wild type. The take-home point was that the changes in cyclic AMP were relatively minimal between untreated cells and compound-treated cells, but there were some trends that suggested that possibly these compounds could have an effect. And as Dr. Fierro had mentioned, one of the concerns is that the assay that we use, the chemical assay, may have some technical limitations and may not necessarily be the best assay to really say whether these cells inhibit the downstream activity of this protein. And one idea that we had was we identified that even though for some of the cells, there can be some changes at baseline between cyclic AMP levels in the wild type and the mutated cells, when these cells are stimulated with an agonist or treated with forskolin, which increases adenylyl cyclase activity, there's a much bigger difference in the R201H, R201C cells in that they make just a lot more cyclic AMP. And so for many of the, to test these compounds, sometimes if you do agonist stimulation and then you inhibit, you can detect changes a little bit more easily. The system can be more sensitive. And so that's what we're working on right now using our iPS cell model. We're testing these same compounds based on different types of drug stimulation. And we were excited to report that this, this project here makes up one of the aims in an R01 that was funded and comes out of a collaboration with Dr. Fierro, Dr. Shao, and Dr. Niesensen as well. So this is kind of an exciting potential next step. The other question that we were trying to answer was whether these compounds directly bind the protein itself. They were predicted to bind, but we didn't have any direct evidence that they did. And the plan is to use surface plasmon resonance to actually measure the direct interactions between the compounds and the protein. As it stands right now, we haven't been able to perform this work. In part due to supply limitations on the compounds and building up that and generating that amount of protein. But we are in the process of trying to look into this further as well. So in summary for this one part, I think that what we've shown here is that our MDBR funded research has led to successful future grant funding and has really allowed us the space, the time, and the ability to collaborate with others on exploring the role of these specific compounds. And as you heard from Dr. Hoffman and Dr. Fierro, they will also be testing these compounds in their system, and we're working on optimizing other systems as well to better detect whether these molecules can have an effect. And just to emphasize the ongoing strong collaborations that have come from this work. And then very briefly, this was presented at the conference back in September, but for those who were not able to attend, I wanted to touch very briefly on the project that was funded earlier by MDBR. That was a joint collaboration between myself, Dr. Shao, and Dr. Fierro. And that's looking at the single-cell transcriptomics to better understand fibrous dysplasia of the bone. And so we understand that the GS-alpha overactivation in bone is what causes fibrosis and abnormal bone formation. And we believe that that is due to the presence of the mutation in the skeletal stem cell. But we still don't know entirely what cell types carry the mutation and what cell types are responsible for the pathophysiology, which is really what a lot of our work, and as Dr. Palmisano was describing too, trying to understand the role of each different cell type. And so we harvested bone from several different patients who had FD and went for surgery. And we collected the CD45 negative cells, which are the non-hematopoietic cells, and performed single cell RNA sequencing using 10x genomics technology. We have 4 FD samples and 4 wild type or non-FD samples that were taken from patients with osteoarthritis. And so very briefly, I just wanted to show some of our data. We were able to show that FD lesions contain stromal, endothelial, and smooth muscle lineages. And that the FD bone cell lesions, the cells that come from those lesions have unique transcriptomic profiles compared to the non-FD lesions. And they upregulate fibrotic signaling pathways, as well as multiple other different growth signaling pathways, which is very, very interesting. And there seems to be potentially a shared FD gene expression signature that can exist across multiple cell lineages. So whether the cell is stromal cell population, endothelial cell population, or vascular smooth muscle cell, they do have some shared genes that are expressed across all three different cell types when compared to wild type cells. And when you look at just the stromal cell types, we're able to do a sub-analysis that also reveals the same types of changes across different types of different cell types. And we're able to identify some cell populations, stromal cell populations, that appear to be unique to the fibrous dysplasia samples. So in summary, the funding that was used here to support the early stages of the single cell RNA sequencing has really been a useful tool for exploring the questions of the altered cellularity and transcriptomic profile of the FD lesions. And we have continued ongoing work where we're performing single cell genotyping to try to determine which cells carry the mutation, which cells don't, and look at the transcriptomic profiles of those. And again, our group at UCSF and Fernando's group at UC Davis had separate grants through the MDBR to look at single cell RNA sequencing, and we collaborated together. And from that, we're able to really advance some of this work that we would not have been able to do without the help of the MDBR and without the ability to collaborate together, which I think is just one of the huge strengths in this field. Thank you.
Thank you, Kelly. That was very nice. It's 4:09. I think, Tova, we were going to stop at 4:15. Is that right?
Yeah, if we have a couple questions, maybe two, could we squeeze in?
Yeah, we had a couple left over from before. One was a question from Cindy Brandt. And this, I think, can go to everybody. I think the answer is pretty simple. Is there a need for more bone samples? The answer is yes. What besides funding do researchers need from our community? That's a big, broad question, open to all of us, I guess. I think what is needed and what we're seeing here today is the funding and collaborations that are and the research and the collaborations and the research that are coming out of that. Others, do you have, what do you guys need from the community? Money. So. Well, while you're thinking about that, Kelly Cohen also had a question. Kelly?
Thank you. Which is kind of funny because I was asking about availability of human FD cells, and also, do you use human serum blood samples? And would you need them of the mutation? Any of the researchers? Is that something that could be used for your research?
So I can comment. I think certainly bone cells are helpful. One of the limitations is that it's always better to be able to process these samples fresh. But we have multiple sites, throughout the country, the NIH or UCSF or UC Davis and other centers that can use these. So if a patient is going for surgery, we try to just send them to whatever place send the cells to whatever place can use them fresh. And then we all collaborate with the samples. And I think everyone's been really good about sharing what we have. In terms of the serum, so because the mutation typically is not carried in the cells that are in the bloodstream, we haven't been, our research team hasn't been, collecting blood, but I know other folks have been, and probably banking the serum, which I'm sure could be helpful. But if there's anybody on this call who has active research projects where they're looking at the serum from, the peripheral blood, I'll let them chime in.
Well, we've been collecting serum at the NIH, and we have a lot of stored serum samples, and we would be glad to share them with people who have, sort of well-defined, likely to be successful, or, well-designed experiments that are reasonable to do. Yeah, we'd be glad to share them. I would encourage everybody who can collect blood samples to collect them and save them. I mean, the notion of, extracellular vesicles in the serum that are secreted by bone cells is emerging as, a likelihood. And so if samples are collected appropriately, they could be used for that. So I think it's a great idea, serum samples, Kelly. Kelly, why did you ask that, I wonder?
I asked that just because I would assume that it's more available, easier to get. And then also, I was going to ask if you can use, well, obviously, I'm sure you could, cord blood samples, and I'm sure that's extremely rare in this community, if there are cord blood samples.
What kind of blood samples?
Cord blood, like
cord blood samples.
birth. Because I'm cultivating, and I'm hoping that will do something with his cells. And is it something that we're asking people if they have with their children?
I don't know. So we've got 1 minute left, and maybe we'll just use this to close and thank everybody, and especially thank all the presenters. I think all the presentations were really clear and straightforward, and that's not always the case. I think many of us have gotten better at speaking to sort of mixed audiences over the years, and that really came through. They were really great presentations. And again, I think the use of the funding from the $1 Million Bike Ride, it's, I think this is an amazing example of the success of a program like this. And thank you, Cindi, for doing this over the years, and thank all the presenters. Sarah has something to say. Sarah Mumper, I see you have your hand raised. Hi, Sarah.
Hey, everybody. I just wanted to kind of make a comment as a patient and who's involved with a lot of the patient groups. I know how important it is to get bone samples, especially just being part of NIH and being connected with this group. However, I don't think it's really accessible to a lot of the patients who aren't necessarily in the know or aren't connected with doctors or researchers in that way. So I just kind of want to put it out there that perhaps there's a better way for us to do outreach to those patient groups, whether it's how to send these samples, how to talk to their surgeons to provide those samples to the researchers. Just something to keep in mind, that from a patient perspective, I know those things, but not the majority of the groups know those things.
Yeah, I think that's a great idea, Sarah, and I think that is a barrier, and it calls upon the collaboration of the doctor who's operating on the patient. And frankly, for many of them, it's easier not to do it. The rules that have been put in place to protect patients which protect the patients, but they've created a tremendous burden to doing some of the sharing that used to be easier in the past. But I do think, I think you point to a good thing, that perhaps the foundation could help put together a sort of information package, and maybe in the future a sort of real package of ways to send this that could be helpful and make this easier in the future. Cheat sheet for collecting samples. That's a great idea, Don just put in the chat. Yep. And frankly, at some point, it does require sometimes putting some pressure on your doctor to do that, which is easier said than done sometimes.
Thank you
We run a
so.
couple minutes over, Tovas, but so you want to?
Yeah, no, I'm so grateful to everyone. Thank you so much to our presenters and to everyone for being here and part of this conversation. I do want to just make a quick plug for our upcoming programming. We have, I mentioned earlier that we've had some great success through the Department of Defense peer-reviewed medical research program. Researchers who've previous $1 Million Bike Ride awardees have gone on to secure funding, and we have an information session coming up for that on the 19th. And then also a caregiver social happening later this month. And all of that, you can respond or register for that on our website. Thank you so much. And please do feel free to reach out if you have further questions.
Can I make one final plug? There will be another international craniofacial, international FDMAS meeting in Madrid in the fall of 2025. So that should be really great. The one in Silver Spring last year was a great success. So everyone's keep that on your radar.
Mark your calendars. Absolutely. And yes, if you do plan on submitting an LOI for the PRMRP, please let us know. Thank you, everyone, and be well.
Thanks. Bye-bye.
Thanks, everyone.
Thank you.
Bye.
Bye.
Thanks. Bye-bye.
I'll see you, Adrian. Hello.
Bye.
Hey.
Thank you
Bye,
so
Tova.
much.
Thank
Bye.
you.
Take care.
Ciao, ciao.